My talk today will be about a project that I started many, many years ago, a project that actually has gotten into an app that you can use online. So if you would like to register and get an account and see what it does, it's called ResearchMaps. You'll see why it's called ResearchMaps. All you got to do is click on this thing here. Why all of this? Essentially, this is a problem that my lab faced. This problem that Peter already talked about, the immensity of the published record. 25 million papers is a number so high that it no longer makes any sense to us. In my own field, so 100 million experiments, at least, probably closer to 200 million experiments published. You will hear a lot of big data problems today. This is undoubtedly one of the most profound ones for which we have done very little to address. How do we deal with a number of 100 million experiments, right? So if you go into a particular field like my own, learning and memory, you're talking about at least 1 million experiments. How do we organize all of this to make sense of it, and more importantly, to decide what to do next with all of this? In synaptic function, another area that we work on, at least 3 million experiments published, right? This is a recent problem. Why is it recent? Because of this incredible growth in the literature. When I was a graduate student, this wasn't as much of a problem as it is now. You know, technology has gotten far more complex. Individuals, neuroscientists are asked to work in a variety of fields. All of this is relatively recent, and we have not caught up to develop tools to help us deal with this, complexity and diversity. Now, what we decided to do is to build maps. Of course, when we have lots of stuff that we don't understand, one way to go about it is to map it. Map it organizes it. So what do I mean by maps? So imagine that you would like to know something about neurofibromatosis and learning, and you go to PubMed and you get a bunch of papers out of PubMed. You could spend a week reading them carefully. Let's say you were going to do work on this area. Or if there were a map, such as this cartoon here, you could simply look at it and know that NF1 inhibits RAS, that RAS is needed for promoting MAP kinase, that MAP kinase regulates GABA release, that GABA release modulates plasticity, and that plasticity is involved in learning and memory. And then you could see by these numbers that some of these connections are more dependable than others because these numbers are higher and lower. And then you have these little symbols here that tell you about the kinds of experiments that were done. So instead of spending a week, you could look at this map and quickly have a sense for how much work has been done in this area. Now, that's a thumb sketch of what I'm about to tell you for the rest of the lecture. So what is a map really? Now, a map has several things. One of the things that it has is phenomena. In science, we make webs of phenomena, A causing B, B causing C. That's one of the key things that we do in science. Not the only one, but one of the key things. For example, in this case, Crab and memory. My own lab has worked on this. So these are phenomena that my lab is interested in. And work in my lab and other labs show that Crab promotes memory, so that there is an excitatory connection between Crab and memory. So you have connections in these maps, you have phenomena. Of course, there are different kinds of connections. Not everything promotes something else. Every time you have a green light, you have a red light, an accelerator, a brake. So in our own work, we know that PPI, this phosphatase, is involved in regulating memory. So more PPI leads to blocking of memory. So inhibitory, excitatory connections, they're both in maps. And of course, another thing in these maps is that you have connections, you have the evidence for no connection. So you have excitatory, inhibitory, and evidence for no connection. And then you have weights, because things are not connected or not, things are connected to different degrees of evidence, right? Sometimes we are uncertain of whether Crab really has a role in memory. So I thought that the maps needed to have a number that reflected that kind of information. And the other thing that's critical is that the map should also have symbols that tell us about what kinds of experiments were done. And I'll tell you about that. So in this case, we were talking about 4 different types of experiments that I'll describe. So you have phenomena, we have connections, we have weights, and we have experimental symbols. And the idea is that with this information, you will have a sense for what's in these papers that you've just read. So essentially, this is, again, a very brief summary. And what we have done with the app is to automate all of this. So you can read a paper, place this information in, and then get a map out of it. And eventually, we are currently working with Klaus Müller with machine learning to automatically populate this map based on what you have done by hand. So you put in a paper, and hopefully machines will put in 100 or 1,000 or 1,000,000, depending on how much data you have put into the app already. So that's what's coming in the future. By the way, the app is an UCLA-based service. It's completely free. There's no companies associated with it. It's our app. If you are a programmer, you would like to participate in it, please let me know. It's open code. Everything is shared. If you have something that works best, we will substitute it and keep on running it. So that's the idea, and that's what this is. Okay, now what goes on inside of our app? So let's say you start with a paper, you translate this paper into statements that are key to what the abstract claims in the paper. So the experiments, you distill out of the paper the experiments, you enter them into the app in a very simple way. It takes me about 10% longer to read the paper carefully versus reading and putting it in the app. So it's really not a lot of investment. So you put it in the app, and the process of putting in the app actually helps you understand what the paper actually says versus quickly reading, getting some sort of intuition over it, and not really knowing what the paper says. But if you put it in the app, the paper is that important to you because you're going to write a grant, you're going to write a paper based on it. The process of putting it in the app is very reinforcing. Now I hate reading papers that I really want to know and remember without putting them in the app because that process is actually very generative. Then the app essentially sets up a relational database of this information, and I'll tell you how, and then the app graphs it, okay? So we went with Neo4j. Before we tried a classic SQL database. I think we like this better because it's faster, more appropriate for the kinds of programming that we do. And this is what the app looks like and one of the maps that we got. So, okay, maps. What is it that we need to build a map? In building a map, you need at least two different types of things. You need a strategy to classify experiments, right? Because this is what we want to map. We want to map experimental findings. And the other thing that you need is a set of rules for drawing those maps, right? So you have to figure out what is it that you're going to map and then how you're going to map it. And that's what I'm going to tell you next. Because the app, without understanding what goes on in its brains, it's utterly useless. But what I hope to do in the next few minutes is to convince you that we didn't invent any of these principles. All we have done is to implement principles that we all work and use in the lab. All we have done is to automate them now so that a lot of this can be done without us really having to do it by hand. Okay, so the first topic then is to try to focus on a strategy to classify experiments into categories. Let me start with a disclaimer. It's a light disclaimer, but one nevertheless. In neuroscience, there are many areas. Our work is on molecular and cellular cognition, which is an area defined by connections between molecules, physiologies, and behaviors. That's the bulk of what we do. So the app and all what's behind it is really focused on any studies that connect between levels of analysis in neuroscience. However, I should say that we have done a lot of work that shows that molecular and cellular biology in general, even cancer, development, immunology, all of that plays by the same rules as molecular and cellular cognition. So you could easily use it for all of that. Even behavioral neuroscience, if you study diseases and aging, this may apply and be useful too in development in terms of neuroscience, development neuroscience, or otherwise. Okay, so this is to say, for example, that systems neuroscience and cognitive neuroscience, I'm not sure whether you can use the app as well as for these other areas. You can, but that's not what was designed for at this point. And if you would like to help in the process of including those areas in the app, will be wonderful. Okay. So what about the taxonomy? Remember, if we're going to map, we have to map experiments. If we're going to map experiments, we have to divide them into groups. We have to develop a taxonomy. So in molecular and cellular cognition, there is at least 3 different types of experiments. And we have thought long and hard for the last 15 years about this. And we can only think of 3 different major categories. In one category, identity experiments, you essentially define the things that you're interested in. In the second one, connection experiments, you connect them. So you define the things you're interested in and you connect to them, Crab and memory, for example. And then tool development experiments, where you develop the tools, the approaches, the methods that allow you to do identity experiments and connection experiments. Now, we focus our early work on connection experiments. And when we looked at all the experiments in the literature, we have now placed almost 2,000 experiments into the app, just a drop. We found out that we could divide all of those connection experiments, where there's an A and a B, I connect A and B, into 3 different categories. Positive manipulations, where you increase the levels of A and look at B. Negative manipulations, where you decrease the levels of A and look at B. And non-interventions, where you don't manipulate A and B, you simply look at them. So these are the 3 different types of experiments. And this just, again, repeats what I said. Positive manipulations, you increase A, look at B. Negative manipulations, you decrease A and you look at B. And non-interventions, you look at A and B and see how they covary. Now, it turns out that besides single connection experiments, you also have multi-connection experiments. So these are experiments where you don't just have A and B. You have A, B, and then C, where you manipulate A and B and then you look at C, for example. Okay? In this case, they're slightly different, but the idea essentially is that in single connection experiments, you have a single agent and then a target. And in double connection experiments, you actually have 2 agents, where you manipulate NMDA receptors and chymosine kinase 2, and then you look at CA1-LTP. The idea being to see if chymosine kinase 2 mediates the relationship between NMDA receptor and CA1-LTP. These experiments are very, very common in science, and they are represented in our app and in our taxonomy. When you look at identity experiments, there is at least 3 different things that define any entity that we are interested in. There is the what, which has to do what that entity is, and its composition and its interaction with other entities. There is the where, that defines where that entity is. Is it in a spine? Is it in the nucleus? Is it in the hippocampus? Is it in the hippocampal formation, the cortex? So this is relevant information of where that entity is and when. Is it early in development, late in development, etc. Putting together these 3 things can define unambiguously just about anything that you study. And that's critical. If you're going to connect things, you must know what those things are, right? And where they are and when they are. All right. So, again, what I already just said, what, where, and when defines individual phenomena that you are interested in. Now, so here you have the whole, taxonomy put together. So identity, tool, and connection experiments, and then multi-connection experiments I talked about, positive manipulation experiments, negative manipulation experiments, non-interventions, and the 3 ways in which you can define identity experiments. So why am I bothering you with this? This is essentially the things that we mapped in our web. And what I'll propose to you is that if you want to keep track of the complexity in science, you would have to at the very least, care about these things and look and see how they are connected, right? So if you want to extract valuable and relevant information about the 25 million papers, the probably nearly 200 million experiments in PubMed, this is a good place to start. Is this all inclusively? Certainly not. We focus on molecular cell cognition, on molecular cell biology. There's far more to biology than that, but this is a place to start. And I think with this framework, you could probably include other things that are more relevant to the research that you do if it happens to fall outside of this area. The second thing that I want to tell you, so once you decide what you are going to map, and there's a categorization of those things that you want to map, the next thing is to decide a set of rules to draw those maps. And I won't have time to tell you about all of these rules. I will only focus on 3 of them very quickly. But this is critical because these rules that I'm about to share with you are the rules by which we generate these maps and then implement it in the app. So the first rule is very simple. The first rule is convergency analysis. Again, remember, we didn't invent any of this. We just formalized what people like myself and you do in the lab every day. So what this shows is that if you have evidence that's convergent from different types of experiments, positive manipulations, negative manipulations, non-interventions, and then mediation experiments, that this connection should have a bigger weight than if you have another connection that's supported by just one type of experiment. Okay? So convergency is critical in science. We all know this. We practice this in our labs daily. So the other thing is consistency, which is also critical in science. What does consistency mean? It means that if you have a connection where you have two lines of evidence that are consistent, that connection should have more weight than than another connection that has just one line of the same type of evidence. So convergency, different things telling you one story. Consistency, multiple times the same thing telling you the same story. Of course, if you have violations of convergency or consistency, then instead of getting more evidence, you have less evidence for that connection. Instead of having this connection having a greater weight than this one, now if there's an inconsistency here, then this experiment will annul this one, and then this connection is not as valid. We can't trust it as much as this one. Right? So again, I didn't invent any of this. I'm just formalizing it. And finally, multi-connection analysis, and this is very easy, which is if you have two connections, in one you know something about the mechanism of how A gets to B, this connection is more trustworthy than one for which you have no mechanistic insight. Again, this should feel familiar to everyone in the audience because this is what we do every day. All right, so how did we use all of this? Let me just skip through this. How have we done all of this? What have we done with all these rules? What we have done with these rules is to develop a system to weight these connections so that when you look at the map, at a glance, you immediately know something about the level of evidence for the things that you're interested in. So let me go quickly through it. One of the first things that we did is to look at this table here. This is experiment 1, experiment 2, 3, 4 of the same repetition of positive manipulations. So for the first experiment, we give it a weight of 0, 1 to 5. Why 0, 1 to 5? You'll see in a second. Because we have no reason to say that these experiments are more important than this one, or this one more important than this one, or this one more important than that one. We just gave them all equal weight. You know, perhaps eventually when we look at the history of science, we may find evidence that one is more valuable than other in one domain, and in a different domain that will be more valuable than other, more predictive than other. But at this point, we don't have that information, so we gave them all equal weight, okay? Now, we know, so that's essentially it. So you can see that if you're looking at this connection, you have 4 different lines of evidence for it, this connection will have a very high weight of 0.5. However, the second time you do a positive manipulation, we know that that's not as impactful as the first time. So we simply gave it a geometric series to it. So the first one is 0, 1 to 5, the second one is half of that, and then the third one is even half, and so on and so forth, okay? Now, of course, if you have the opposite, if you find out that actually a positive manipulation gives you a conflicting result, that there is no connection there, you do a positive manipulation of A, nothing happens to B, well, that's the beginning of an alternative hypothesis, that A is not connected to B, because you manipulated A, nothing happened to B, so you simply take away the weight that prior experiments provided for how A is connected to B. So you saw that this weight went down from 0.6 to 0.4. No one experiment is definitive in science. In science, is really an accumulation of experiments that tell you about the picture in both ways, in increasing the reliability and the trustworthiness of those experiments or decreasing it, right? And essentially, that's what the app does. It's essentially implementing principles that you and I do all of the time. All right, so what is the dream? The dream is that eventually a lot of this process will be automated, and that every time there's a new paper that comes on, it is automatically added to this enormous web of science, where our assertions about the identity of phenomena, our assertions about the connections between these phenomena, get automatically embedded into a very large network that allows us to query and interact with this massive amount of information. Is this all that science is about? Of course not. There's much more to science. It's just a tool. But think about the time that this will save us. So what our app allows us to do is to take a network, not as big as this one, a tiny little network, but still already we have 2,000 experiments. The network is far too complex for you to grasp, remember, and hold in your mind at any one time. But what you can do is that you can query that network and interact with it and see the complexity in it that's useful to your own work. That's the value of it. So without further ado, I would love to answer some questions. Thank you.